All right, hello everyone. I hope you all are doing okay. I chose to focus my grant on Alzheimer's disease, but specifically I was looking at the post-translational phosphorylation of tyrosine 18 and threonine 231 and tau in the earlier stages of Alzheimer's disease. The symptoms and pathology of Alzheimer's disease. So Alzheimer's disease is a leading cause of dementia. Dementia presents with a variety of symptoms, including alterations to cognitive function, the loss of memory, loss of executive function, and changes to behavior and personality. This results in an inability to perform the basic activities of daily living, and you can't live on your own. Alzheimer's disease presents with a couple unique symptoms. In the earlier stages, the patients present with anxiety, depression, and withdrawal from social interaction and from other people. In the later stages, there's aggression, poor judgment, confusion, and sometimes the presence of other neuropsychiatric disorders. In diagnosis, there were criteria that are determined by the National Institute on Aging and Alzheimer's Association. This was created to help standardize the diagnosis of different neurological disorders. For dementia, as I mentioned before, there's the inability to function in daily life, decline in performance, no prior psychiatric disorders, and cognitive impairment. More specifically for Alzheimer's disease-related dementia, there's a gradual onset of symptoms over months or years, so it takes a really long time for these symptoms to occur. There's major impairment of language and executive function, and lack of symptoms of other neurological diseases like Lewy bodies, which are associated with Parkinson's disease, or aphasia, which is the loss of speech abilities due to brain injury, like getting your head hit or being in a car accident. I went pretty in depth with this in my grant, but there are a lot of common testing tools that are used for diagnosis. Serum tests, brain MRI, CT scans, cerebrospinal fluid tests, genetic testing, and amyloid PET scans are all the most common ones. These all increase in cost and complexity as you go on and get tested. Here they're looking for pretty much any abnormality, especially phospho-tau, amyloid plaques, atrophy, and neurodegeneration. Unfortunately, we can only diagnose with 100% certainty post-mortem, but these tests can help you determine with like 95% certainty. I thought this was a pretty good diagram to show what happens in the brain and different cells in the brain during Alzheimer's disease. The brain atrophies, so it kind of shrivels up a little bit. There's the plaques and the tangles, and then the different cells change their morphology. And in diagnosis, there is BRCA staging. This uses the presence of phospho-tau to diagnose patients. There's an antibody called AT8 that binds to phospho-tau strictly, and in these different stages, it increases in both presence and it increases in the different locations. So in stages 1 and 2, it's in the entorhinal cortex. 3 and 4, it's in the hippocampus and limbic system. And then 5 and 6, it presents in the frontal and parietal lobes. I'll go more in depth with this later, but these are the general stages. So the current standard of treatment, the goal is to slow the onset of Alzheimer's disease, and there's a couple of medications that patients can take. Cholinesterase inhibitors prevent the breakdown of acetylcholine, which is needed for like muscle function, and it's used throughout the body. And in Alzheimer's disease, it's broken down way too fast. NMDA antagonists interact with glutamate to help with its transmission, and both of these drugs are pretty well tolerated. Sometimes there's GI tract issues and nausea, but other than that, there aren't any severe side effects, so they're commonly used. There are in-home options as well. Families can take care of patients or they can go in a nursing home, but unfortunately there is no cure for Alzheimer's disease. These are just measures that help patients cope. So the possible causes, modifiable risk factors are anything that you can control. So you can work out, eat well, you can read and do crossword puzzles and that kind of thing just to keep your brain strong. There's also non-modifiable risk factors, which are things you can't change. Your age, family history, if you have a genetic disposition, anything like that, you can't change. It's just something that you have. With familial Alzheimer's disease, this is a lot less common, but it presents with abnormalities in penicillin 1 or 2 and the amyloid precursor protein. It also presents much earlier with early onset. This is classified as anything that appears before the age of 65, but more commonly it appears in your 50s, sometimes later 50s. Sporadic Alzheimer's disease is much more common. This is because it's just caused by random mutations that occur. The vast majority are caused by mutations in the ApoE4 gene. This causes increased amyloid deposits and earlier onset similar to familial AD. The other leading cause is hyperphosphorylation of tau, which causes tau tangles. I thought this was a pretty cool diagram, but this shows the formation of tangles on your right and formation of amyloid plaques, and either of these, they mess with the morphology of the cell, which causes the neuron not to be able to function the way it's supposed to. So tau. It is a microtubule stabilizer in the brain. It promotes a whole bunch of key functions within the cell, but some of them include neural outgrowth, transport along the axon, and membrane interactions. It's key in microtubule binding and assembly, and it helps regulate vesicle transport because it plays a part in anterograde and retrograde transport, which is transport towards the cell membrane and away from the cell membrane. It helps with synaptic activity and plasticity. This is the continued growth and changing of neural synapses, which is key in memory and learning and be able to do new things. It's encoded by the MAPT gene on chromosome 17q21. This gene can undergo alternative splicing, which produces alterations in 3' and 4' tau. 4' tau binds a lot tighter to microtubules and helps with its interaction more so than 3' tau does. Lastly, the microtubule binding domain is on the C terminal of the protein, and this is what actually interacts with microtubules and gives tau its key function. You can see in this diagram the formation of these aggregates that it interacts with the microtubule, but not in the way it's supposed to, which is why the microtubules no longer function. So some common mutations and modifications. The vast majority of alterations to tau are post-translational, but you can obviously have missense mutations in that codon region that cause similar effects. Alterations in exon 10 splicing, which I mentioned in the previous slide, causes a skewed ratio of the 3R to 4R tau. Because 4R tau binds so much tighter, if there's too much of the 4R tau, then the tau just does not function. Also, if there's not enough of the 4R and there's too much 3R, then it just won't bind at all. The improper acylation and hyperphosphorylation both present with the same physiological tau tangles, but is obviously from different causes. These taupathies that they cause produces really filamented tau, and this filamented tau is what actually induces the formation of the tangles. So in Alzheimer's disease, tau is up to 3 times as phosphorylated as normal tau, which is like an absurd amount. Normally we see this with the initial hyperphosphorylation that causes the aggregation, and then the tangles form, and then that's the alterations to the tau function. I'll be going a little bit more in depth with the Braak stages here. So we have the entorhinal cortex, which is located at the intersection between a whole bunch of memory structures in the brain, which includes the amygdala and hippocampus, a bunch of other structures that I don't know how to pronounce, so I'm not going to say them. But all of them are really critical in memory, and it does function as a bridge between the hippocampus and other structures in the brain. We then have Braak stage 3 and 4, which is the hippocampus and limbic system. The limbic system includes the hypothalamus, hippocampus, fornix, and amygdala. This all plays a role in memory, emotion, learning, and motivation. Lastly, we have Braak stages 5 and 6, which is the frontal and parietal lobes. Any phosphorylation here has drastic change in behavior and executive function. There was a pretty good question on the discussion boards about the phosphorylation stages. So what happens with the grading by Braak stage is that if you're in Braak stage 4, for example, you do have the phosphorylation in the entorhinal cortex as well as the hippocampus and limbic system. So the later stages are inclusive of the earlier stages, if that makes sense. So some key phosphorylation sites within tau. Serine 202, threonine 205, best characterized, and they're highest in stages 5 and 6. I'll be calling these the later sites. Serine 199, 422, and 396 are all less characterized. They're also highest in stages 5 and 6, and I'll also be calling these the later sites of phosphorylation. That's because these later sites, I'm talking about them in relation to these two sites, which are the focus of my grant, threonine 231 and tyrosine 18. These present in stages 1 and 2 and increase through stages 5 and 6 of Brax staging. These are actually the only sites that have extensive phosphorylation in stages 3 and 4, so you can really see these sites being phosphorylated in the limbic system and hippocampus. This is actually thought to be some of the earliest events in the progression of Alzheimer's disease, with the phosphorylation of threonine 231 specifically being maybe the first, if not the first, it's in like the first few stages of Alzheimer's disease. Lastly, from my background, some key regulatory kinases. GSK3 beta is important in a bunch of signaling pathways. It plays a role in neural development and neural migration and axon outgrowth. I'm interested in it because of its ability to help rearrange microtubules. Its inhibitor is abemaciclib, which is currently FDA approved to treat cancer, but it interacts with tau, which is why I'm interested in using it here. CDK5, cyclin-dependent kinase 5, functions in the central nervous system. It's regulated by P53 and P35, and when a cell is under stress, it cleaves P35 into P25, and this P25 actually causes CDK5 to become constitutively active. Its inhibitor is phenytoquinolone derivatives. Not sure if I pronounced that right. It's not FDA approved, but it has been shown in several studies to prevent the phosphorylation by CDK5. Lastly, we have MARC, which is microtubule affinity regulating kinase. This is important in regulation of vesicle transport and cell migration. It actually functions really powerfully in the hippocampus. Its inhibitor is silcyclohexyldiamine derivatives, which, like CDK5, it's been shown in numerous studies, but it's not FDA approved. Lastly, I have protein phosphatase 2A. This is a potential regulator of these three kinases. If you didn't know, phosphatases take a phosphate off of a protein or structure, whatever is phosphorylated. So it's believed this is a pretty important regulator of these sites. Okay, let's get into the science. I have three aims, but my first aim is to identify the key kinases that I mentioned that play a major role in the post-translational hyperphosphorylation of tau and their inhibitors. Then I plan on identifying the kinase that phosphorylates threonine 231 and tyrosine 18, as well as timing in relation to the other sites. Lastly, I plan on determining if the hyperphosphorylation of tyrosine 18 and threonine 231 are necessary for the phosphorylation of the later sites that I mentioned. So aim 1, research design. Here's my aim if you have forgotten since the last slide. But the first thing I want to do is kinase overexpression or knockdown in the NT2 cells. The general steps that I'm going to do are transfection, flow cytometry, and then Western blot. So I chose the NT2 cells. This was a question I got a whole bunch of times in the comments. This is a brain tumor cell line. It retains some stem cell characteristics, but it only differentiates into neurons, which is why I'm interested in it, because I only want to work with neurons. It also allows me to make a stably transfected cell line so that when I transfect the kinase in, the future generations of the cell line will keep that kinase in its genome. So how I plan on doing this is that I will transfect with shRNA for knockdown through the calcium phosphate method, and I will overexpress the vector with a CMV promoter for pretty high expression with the calcium phosphate method again. I will also have cells that have a normal expression as my protein control and have an empty vector as my transfection control. I will then do flow cytometry to make sure that the cells are actually alive, which is really important. And I will then perform a Western blot to determine the baseline amount of protein within the cells, and then I can compare this to the knockdowns and overexpressions to make sure that the transfections that I performed were actually successful. Next, I'm going to purify and analyze the tau phosphorylation levels within ELISA. So initially I'm going to purify tau with immunoprecipitation. This method of precipitation actually produces a whole bunch of different fractions. So I'm going to use UV analysis to decide which one of the fractions has the most protein in it, and then I will pool the top 3 fractions together and run an ELISA. For the ELISA, I will be doing this to quantify the amount of phosphorylated protein using a phospho-specific antibody, and this should tell me which kinase is most active in vivo.